Hello, my name is Andrew Chen, and I'm currently a postdoc at the George Washington University. And the title of my talk is Imaging Therapeutic Effects of Ultrasound on Human Islets in Vitro. So first, I want to start with a little motivation about this work. Our lab has a special interest in using therapeutic ultrasound to combat endocrine diseases, specifically with a special interest in type 2 diabetes. And there are some interesting reasons for this. The first being that ultrasound's potential to modulate endocrine activity remains largely untapped across endocrine organs, but with special interest to the pancreas. Additionally, pharmacological treatments of type 2 diabetes, which is the current standard, have their own set of side effects and may actually accelerate disease progression in early stages of the disease, meaning that ultrasound may present an interesting alternative to the typical pharmacological treatment in terms of mobilizing the natural calcium stores. So this is just a bit of background on the pancreas itself. The organ sits just below the stomach and is nestled kind of in the notch of the duodenum. And the main pancreatic duct, which carries the exocrine secretions, empties into the duodenum via a hole called the ampulla of Vater, which is shared with the common bile duct. Now, the endocrine pancreas is responsible for secreting insulin, and the endocrine cell mass of the pancreas is actually only about 1%. That's mainly islets, or that's mainly beta cells, which are aggregated into islets, or kind of small clumps, also known as islets of Langerhans. On the right-hand side, we see a single beta cell and how glucose-stimulated insulin release happens. For the purposes of this presentation, the important step to remember is that calcium influx triggers a release of insulin from insulin-filled granules from within the beta cells. So in a series of studies performed by our lab, we first showed the effect of ultrasound parameters on the viability of beta cells, specifically a rat insulinoma model. We tested 4 ultrasound frequencies at 1 megahertz, 800 kilohertz, 600 kilohertz, and 400 kilohertz. And we showed that up to 35 minutes after treatment, 2 of these parameters, 1 megahertz and 800 kilohertz, did not cause a significant decrease in cell viability. Further, when we looked at insulin release due to these ultrasound parameters, we found that 800 kilohertz of ultrasound induced a significant release of insulin when compared to glucose or sham-treated cells. So for us, this was great news because we have an ultrasound parameter which does not cause increased cell death, but also can induce a release of insulin. In a series of follow-up studies, we showed that this mechanism appears to be at least somewhat dependent on calcium. So on the left-hand side, we can see what the experimental setup looks like, where ultrasound is being applied from below this petri dish, which has cultured some insulinoma cells. And on the right-hand side, we can see the detection of insulin by this carbon fiber electrode over time, and that the carbon fiber electrode does detect release during ultrasound stimulation in blue, and then the red trace is the same experiment but performed with the addition of EGTA, which is an extracellular calcium chelator. Essentially, it removes the calcium from the extracellular solution, and the response appears to be somewhat diminished. So this indicates that the mechanism that we're inducing appears to be calcium mediated. So we're interested in observing the effects of ultrasound on human islets for 2 reasons. The first being that this is a more physiologically relevant biological model compared to rat insulinoma cells. And also, using fluorescence imaging, we can probe a number of other mechanisms and dynamics inside of the cells, not just calcium, as we have previously indirectly observed. So this diagram shows how an experiment is typically structured. We receive the islets shipped to us from our distributor, and then we can plate the islets and allow them to adhere for a couple of days. After the islets have adhered to the wells, we can load our fluorescent dye. In this presentation, I'm focusing mainly on a sodium, intracellular sodium and reactive oxygen species sensing dye. And then we can also extract samples for chemical analysis. So at a 0 time point, we can extract some of the extracellular solution for human insulin ELISA. And then we can perform our imaging with ultrasound application, high glucose application, or sham treatment control, and analyze the fluorescence time series data. And then later on, we can take another sample for more chemical analysis. The parameters that we used were 800 kilohertz of ultrasound at 0.5 watts per centimeter squared. And we had a pulsing scheme which essentially prevented too much overheating of the well. This pulsing scheme only induced approximately a 1 to 1.5 degrees Celsius increase in temperature over our 3-minute application period. So this slide here just shows what the human islets look like under a microscope, under a light microscope at the top, and then our confocal microscope in the bottom panels. We can see the bright field on the left, and then the fluorescence channel in the middle. And we can see that the center of the islet appears darker than the edge, which is what this white arrow is indicating. So even though we can get dye penetration into the islet, it appears the center of the islet sometimes has less dye penetration. And we can compensate for this by using a region of interest analysis and looking only at certain parts of the islet, which we know have been loaded well and are giving good fluorescent signal. So this slide shows our sodium fluorescence imaging data thus far. We had 22 trials of ultrasound application and 10 trials of sham-treated human islets loaded with sodium fluorescent dye. And we can see the green and red arrow indicating the start and stop of ultrasound application. Now, over 12 minutes, we were able to induce essentially 1.7% increase in fluorescence on average with ultrasound application. And with sham-treated islets, we saw a approximately 3% decrease in fluorescence over 12 minutes. We can also notice that during the period of ultrasound application, we significantly seem to increase the rate of sodium influx into the cell. These two figures on this slide show the average traces of reactive oxygen species dye. And so what we see here again is that though we get an increase in the amount of reactive oxygen species inside the islets over 12 minutes, we seem to induce a much greater increase of reactive oxygen species with ultrasound treatment. And again, we can notice that between the 3 and 6 minute mark, there appears to be a pretty significant change in the rate of ultrasound, the rate of increase of reactive oxygen species when compared to the sham treated. So from these results, what we can say is that ultrasound does appear to be increasing the intracellular levels of both sodium and reactive oxygen species. Sodium also does play a role in the insulin release, and reactive oxygen species, though that may sometimes be an adverse effect. In certain instances, reactive oxygen can be a stressor for the cells. It's known that during normal cell metabolism, there is also some reactive oxygen species increase. So that may be a result of just the gambit of effects that ultrasound may have on these cells. I mentioned also that we can extract samples for ELISA analysis. So while we are still extracting samples and analyzing samples. This chart here shows the data thus far. Though none of these differences are significant, we can see that ultrasound does appear to be inducing a release of insulin from these human islets greater than sham-treated cells, which is trending in the direction we expect. Thank you for your time.